Hello and welcome to the chapter on the special senses. In lesson 1, we will be considering olfaction and gustation. And in lesson 2, we will look at vision, audition or hearing, and equilibrium. The special senses include your sense of smell, which is olfaction, your sense of taste or gustation, vision or sight, hearing or audition, and equilibrium or balance. The receptors and structures associated with your special senses are concentrated in the head, and the receptors and pathways are anatomically distinct and rather complex. We will be giving you the simplified version of this discussion and an overview of the special senses. The general senses, which is your sense of touch, pressure, pain, proprioception, vibration, tickling, itching, etc. Those are somatic senses that have very basic pathways, so somatic sensory neuron associated with receptors that transduce the signals into the CNS directly along a single neuron. And those receptors are located in the muscles and skin peripherally, while the receptors here for the special senses are located within the head, closely associated with the CNS itself. We will begin with olfaction, which is your sense of smell. The epithelium that contains the olfactory receptors is located in the nasal cavity, covering a region of the ethmoid bone called the cribriform plate. The epithelium contains from 10 to 100 million receptors. That's a lot. So it's a very fine-tuned sense. The receptor cells are bipolar neurons, and the dendrites act as the receptor component, and the membranes branch into tiny cilia that have the appearance of hair, so they're called olfactory hairs. Within the olfactory hairs are receptor molecules that bind to the stimulants of olfaction or odorant molecules. This is a chemical that binds to the olfactory hair receptors and creates a generator potential that leads to an action potential along the axons of the sensory neuron. Surrounding, supporting, and replacing the olfactory receptor cells are basal cells, and these differentiate into new olfactory receptor cells that replace the old ones every month. So that is unusual for a neuron or neuron receptor that the structures are replaced every month, but that's how it is for your sense of smell. I encourage you to enlarge this diagram if you can. You might have to do it after the audio is off, but the story begins with odorants that enter your nasal cavity. They are dissolved in the mucus secreted by globular cells in the nasal epithelium, and the mucus helps to trap the odorant molecules and dissolve them so that they can bind to the olfactory hairs, which act as the receptors. As you can see, those are shown in blue. Those olfactory hair cells are surrounded by and supported by supporting cells, which differentiate into new olfactory cells. The binding of an odorant chemical to the olfactory receptor cell hairs generates a potential that travels along the axons of those receptor cells, which are collectively bundled to form cranial nerve 1, the olfactory nerve. They pass through openings in the cribriform plate of the ethmoid bone and enter the brain in structures called olfactory bulbs. Within the olfactory bulbs, the terminals of those receptor cells transmit the signal to axons of the olfactory bulb that then bind together to form the olfactory tract. So again, now we're within the brain or the central nervous system. So the bundles of axons are called tracts. And these take the signal into the cerebrum, primarily to the primary olfactory region in the temporal lobe, but also send signals to various structures within the brain, including those of the limbic system, so that your sense of olfaction is tightly connected to emotion and memory. So this is your first cranial nerve taking the signal into the brain. It's associated with the cerebrum. It's also the only sensation going to the cerebrum that does not first pass through the thalamus. Keep that in mind. The olfactory receptor cells can bind and distinguish between about 10,000 different odors, and the combinations of those stimuli create the specific smells that we can identify. So the pathway is odorant chemicals bind to the receptors of an olfactory hair, creating a generator potential that leads to an action potential or nerve impulse along the axons of those neurons that make up the olfactory nerve, cranial nerve 1. They synapse with the axons within the olfactory bulbs, and those axons are now part of the olfactory tract that take the signals into the brain and project to various parts of the brain, including the primary olfactory area of the cerebral cortex, believed to be located in the temporal lobe. A related sensation is your sense of taste. So that is called gustation, or the gustatory pathway. Taste is based on the activation of receptors, very similar to olfaction, with a few differences. The receptor cells are associated with a single hair projection from each cell, and several of those cells are bound together to make up the taste buds that are located in projections on your tongue called papillae. We also have taste buds in the pharynx and some on the epiglottis. Taste buds are made up of 3 types of epithelial cells. Towards the outer superficial layer of the taste buds, we have the basal cells that differentiate into supporting cells that surround and again protect the gustatory receptor cells. But these supporting cells eventually differentiate into gustatory receptor cells that only last about 10 days. So your taste bud cells essentially are replaced every 10 days, which is very unique, seeing that these are technically specialized neurons. Taste buds are made up of 3 types of epithelial cells. We have the less differentiated basal cells that develop into supporting cells that surround the gustatory receptor cells. Now, the supporting cells actually differentiate into gustatory receptor cells. So basal cells become supporting cells that become the specialized neural cells of the gustatory receptors. Each gustatory cell has a single hair that acts as the receptor binding site for the taste molecules, and there are about 50 of these cells per taste bud. Taste buds are found in the ridges of the tongue called papillae. Taste buds are also located in the roof of the mouth, in the pharynx, and on the surface of the epiglottis in the larynx. We are able to distinguish between 5 basic types of taste, including sour, sweet, bitter, salty, and umami, which is the taste of savory, or the taste we associate with meat or soy sauce. Naturally, it is stimulated by the chemical monosodium glutamate. You may have heard of it, MSG, or you may not, because while it is naturally occurring, MSG as an additive has health implications. There are different thresholds for each taste. We are most sensitive to bitter tastants, and there's a higher threshold for foods that are sweet. So it takes more stimulants in order to detect sweetness. Tastants are chemicals that dissolve in saliva, very similar to how odorants dissolve in mucus for olfaction. With respect to gustation, as the tastants dissolve in saliva, they bind to receptors in the gustatory hair cells and generate receptor potentials. This leads to nerve impulses along the cranial nerves associated with our taste buds, being cranial nerve 7, the facial, 9, the glossopharyngeal, and 10, the vagus. These carry signals into medulla, where they synapse and then project to the hypothalamus and other structures of the limbic system, as well as to the thalamus, where these signals are sorted before being sent to the primary gustatory area in the cerebral cortex at the base of the postcentral gyrus in the parietal lobe. So to summarize, your sense of smell projects to the postcentral gyrus of the parietal lobe, among other parts of the brain. So like olfaction, your sense of taste can definitely elicit strong emotional responses and memories. Now, when we taste our food, part of the experience is actually due to olfactions, as odors from the food can pass upward into the nasal cavity, where they stimulate olfactory receptors. So when you're tasting your food, you're actually smelling it as well. And if you've ever experienced a cold where your nasal passageways have been inflamed or blocked, you will notice that your food doesn't taste as good. It's really because the taste experience is partially based on your sense of smell. We have the ability to adapt to both smell and taste. While we're very sensitive to smell, we adapt to certain smells within a few seconds and within about 5 minutes to specific tastes. So we're no longer aware of them, or they do not generate conscious stimulation. Here we have a depiction of the gustatory pathway, beginning with the receptors in the taste buds on the tongue and in the epiglottis, the pharynx, and the roof of the mouth, the palate, which project along cranial nerve 7, 9, and 10 to the medulla, where they synapse and then project via the thalamus to the primary gustatory area of the cerebral cortex, as well as to the hypothalamus and other limbic system structures.